The first group of invertebrates that we're going to cover are the cnidarians, sponges, and cnidaria. These are the three earliest diverging groups. If we take a quick look back at our phylogeny for our invertebrates, we have this group on the far right-hand side here. This is going to be the first group that we focus on. These are multicellular organisms. They are all eukaryotes because remember they come out of the Uniconta supergroup, specifically the opisthokonts, and all of those organisms are eukaryotes. All of our animals are going to be distinguished by the presence of two germ layers and multicellularity. You can see that we've got gain of mesoderm here, and then later when we get to our next group, we're going to see gain of mesoderm again. So we start out with two germ layers. We're going to gain mesoderm in a couple of them, and then we're actually going to lose our germ layers in our sponges. The ctenophores are considered the earliest diverging animal group. There are about 200 different species of these organisms, and they are sometimes called comb jellies because they have rows of cilia along their surface. We can see some along this surface here, and those cilia help them with locomotion. So they beat their cilia, it propels them through the water. These organisms have a nerve net. It's not the complex nervous system that we're going to see in some of our leader organisms. And then they also have muscles. They're not quite the same physiology as the muscles that we see in our other organisms. They're actually slightly different. They're basically a single row of cells rather than layers of cells into tissues. So we don't see tissues in these organisms yet. We don't see nervous tissue, we don't see muscle tissue yet, but we do start to see the ability for movement with both the muscles and the nerves. Our next group, the Porifera, or the sponges, are a little bit odd, and students sometimes have trouble figuring out why we consider them animals. The reason is that they actually have genes for a functioning nervous system. They just don't have the cells or the tissues to actually have that nervous system. So they have the genes there, but they're not functioning. There are about 8,000 different species of sponges. Sponges are characterized by a loss of those germ layers. So right at the beginning of our animal group, we have the evolution of germ layers, and then the Porifera group immediately loses them, which means that they have no true tissues. Although they still are multicellular, they do have some specialized cell types that help with their functions. Since they don't have true tissues and they don't have germ layers, they also lack a nervous system. They are asymmetrical. Most of our other animals have some sort of symmetry, either radial or bilateral symmetry. The adults are sessile. Sessile means that they are fixed in place, so they are unmoving. Which is normally a characteristic of animals. Usually our animals are going to be moving, but we still consider them an animal because their larvae are free swimming. So the larvae can move. And we'll see this occasionally in some of our animal groups where some characteristics only occur in part of their life cycle and not necessarily in the whole thing. On the right-hand side, we have a general structure for a sponge. These are a stovepipe or a vase sponge. This is kind of a broad characteristic of shapes for these sponges. You can probably imagine some other sponge shapes. That's just fine. That doesn't mean it doesn't work. This just happens to be the example that we have here. So in our sponges, we see cells called choanocytes. Now these choanocytes are really important for the evolutionary progress of our animals. Those choanocytes are very, very similar to organisms called choanoflagellates, which we saw in chapter 23. So choanoflagellates were individual cells, individual organisms, but choanocytes are nearly identical cell types, except they are part of a multicellular organism. So they don't exist separately outside of the sponge. They are a cell type within the sponge. And so the idea here is that these choanoflagellates could have evolved a symbiotic relationship in which they became a multicellular organism. Choanoflagellates on their own or choanocytes both trap and eat small particles of food that come in through their collar of tentacles, and then they have a flagellum in the center that helps them move. So they sort of direct water and filter water through the center inside of those flagellate tentacles. I'm sorry, not flagellated tentacles. If we look at the sponge structure here, we've got a whole row of choanocytes all along, and so their role in the multicellular organism is the same as what it would have been in an individual choanoflagellate, which is to bring in water and particles for that organism to eat. There are a couple of distinct cell types in these in these sponges, the choanocytes, and then a second cell type called the amoebocyte. The amoebocyte takes in food from the choanocytes and digests it. So the choanocytes basically bring the food into the organism, and then it is transferred to the amoebocytes that actually break it down and can pass the nutrients throughout the rest of the organisms. This is something called intracellular digestion. It's not something that most animals do. This is basically a two-step digestion process where the food comes in one cell and is digested in another. Next, we have the Cnidaria. These are jellyfish, and our jellyfish are going to be kind of our prime example here. But we also have hydras, box jellies, sea anemones, and coral that fall into this category. If you think about most of these organisms, but in your mind, picture a jellyfish or a sea anemone, they're going to have radial symmetry. So if we were to look at a sea anemone from the top, it would be roughly the same all the way around, no matter what plane. We're going to get a mirror image. Same thing with our jellyfish. So our jellyfish have those two germ layers that we see in the early cell types. Early organism types. Most of them have tentacles that are going to help to bring in food, either to detect food or to capture food. So here's our sea anemone food. So maybe a small fish is going to get trapped inside, and then the organism can digest it and bring in the nutrients. All of these are going to be just a little bit different, of course, because they are different organisms. But the general characteristic of the Cnidarians are characterized by a sessile stage and a motile or mobile stage. So we have two different body forms. This is going to be roughly similar to our alternation of generations that we talked about in chapter 6. If you look up the Cnidarian life cycle, it's actually going to look really similar. Basically, one form gives way to the other form. So the Cnidarians contain cnidocytes. These are specialized stinging cells. And within those stinging cells, they have little structures called nematocysts. Nematocyst is essentially the pointy end or the hook end. So the cnidocytes can basically fire these nematocytes, and they reach out and they grab a prey, stick into a prey, whatever it might be. They are activated by a little trigger called a cnidocyl. So these nematocysts might be sticky, they might be stinging, they might be barbed or hook-like to grab their prey, but their function is to be able to catch passing organisms and pull them in. Just like our ctenophores, they have simple muscles and simple nerves. They don't have true muscles. In other words, they're not derived from the mesoderm because they only have two germ layers. They only have the ectoderm and the endoderm. But what they do have is a layer instead of the mesoderm called a mesoglia. And the mesoglia is essentially sort of like a muscle layer. That's where their muscle tissue is. So it's not from the mesoderm, it's from this slightly different layer called the mesoglia. We won't get into the differences in these different types of muscles. Just keep in mind that they're a little bit different than our later animals. In the center of our cnidarian, there's going to be a gastrovascular cavity. This is basically one cavity that acts as the mouth and the anus. So the tentacles or cnidocytes are going to bring in the prey item into the mouth, and then they'll digest it, absorb it, and then any waste will get put out of the same hole, basically.